Hello, my name is Kevin Hirsch, and in this podcast, we're talking about mustard research. The Diverse Field Crop Cluster is an agri-science cluster whose purpose is to support the research and development of 5 high-potential special crops. Those 5 crops are camelina, flax, carinata, sunflower, and mustard. Here to talk about the Diverse Field Crop Cluster research on mustard variety development is Dr. Bifang Cheng, the condiment mustard breeder at Agriculture and Agri-Food Canada in Saskatoon. Bifang, you and I go way back. I was working for Sask Mustard when you joined Agriculture and Agri-Food Canada as the condiment mustard breeder. How long ago is that now?
That's about 15 years ago. I joined the Agriculture and Agri-Food Canada Saskatchewan Research Center in 2009.
Wow.
Time goes fast.
Time flies, doesn't it? As I recall, you took over that program from Gerhard Rackow, if I remember correctly.
Yeah, that's right. Gerhard used to work on condiment mustard breeding, yes.
So when you look at the whole scheme of things, as a dedicated condiment mustard breeder, you're probably a fairly rare breed. Probably the only one in Canada. Maybe you look around the world. How many colleagues do you have in this specific area?
I guess probably I'm the only one in Canada. There might be some mustard breeder in India, but I mean, most people they're definitely canola breeders. There are many canola breeders. Yeah.
Yeah, and in India, it's probably more of an oilseed mustard rather than a condiment, which, of course, the yellow, brown, and oriental that are grown in Western Canada. Now, looking at your research development, and I'm a farmer, and I've used a number of the varieties that you have developed and registered. But looking at the direction of the mustard breeding program, I note that IMI tolerance, in other words, IMI herbicide tolerance, is a priority for the brown and oriental mustard hybrids. Now, that would be similar to the Clearfield program that people would be used to in other crops, a similar kind of herbicide tolerance. Do I have that right?
Yeah, that's correct. That's all the brown and orange mustard. So we created the imicide herbicide tolerant gene plasm. So we're trying to transfer the imitolerant genes from into the Ugura CMS hybrid system to develop the imitolerant hybrid varieties. Yeah, this is a hybrid tolerant. The group 2 herbicide tolerance is similar to Clearfield canola. Yeah.
Now, I guess we should differentiate because brown and oriental mustard are Brassica juncea, whereas yellow mustard is Sinapis alba. So they're pretty different animals to deal with. The IMI tolerance is just in the brown and oriental, at least initially.
Like we also created a yellow herbicide tolerant gene source in yellow mustard, but because there are like a different mutation site in the ALS genes, and so there are different point of mutation. So if the mutation site is different, and then the herbicide tolerance will be different. So in the yellow mustard, we created a group 2 herbicide germplasm with weak tolerance to IMI herbicides, but it's resistant to sulfonylureas. So we are trying to create a new gene source using the EMS approach to create the IMI tolerance as good as in Brassica juncea.
See.
Yeah.
How far along is this work, and will there be any regulatory hurdles with putting herbicide resistance into mustard varieties?
In Brassica juncea, so we already had the approval from CFIA for environmental release of the herbicide trait. So regulatory, there is no problem. So we just did the breeding work. So we have to do some breeding work to transfer the gene to the Ugura CMS male sterile line, maternal line, and the restoring line. So just the time. So there is no regulatory issue. But for yellow mustard, as I just mentioned, that we have a weak herbicide tolerant gene source, and also this is also get approved from CFIA, so there is no issue for release it. But if we want to create a better herbicide tolerant germplasm, and then if we create it, and then we have to do the PNT trial, we have to apply for environmental release from CFIA.
I see. So how, from a producer's point of view, and I know you have to get the trait into varieties that you're going to have the quality of seed there and acceptable yield, and you have to go through variety trials and a registration process. How close is this to actually being available to farmers?
Actually, because we are working on development of hybrids, because it takes time to develop the herbicide tolerant CMs A, B, and R lines. But we are making very good progress. So we already have a herbicide tolerant CMs A and B lines, and also restore lines. And we planted the herbicide tolerant lines in the 2025 nursery. So we sprayed the nursery with the herbicide areas. So all of our restore lines survived the spray. And we also made a preliminary herbicide tolerant hybrids in 2024. So we have some preliminary herbicide tolerant yield trials for the brown and orange mustard. So some of the hybrids showing very good, yield potential, and also herbicide tolerance, and also some, desirable quality traits. So I would say development of the herbicide tolerant brown and orange mustard hybrid varieties is progressing very well. And for yellow mustard, as I just mentioned, that we had a weak herbicide tolerance gene source. So we are trying to transfer this herbicide tolerant gene into different environments to develop composite lines. At the same time, we are trying also using the EMS approach to induce new mutations for the strong herbicide tolerance. But we are not not very lucky. We tried 4 times, but didn't have the desirable herbicide tolerant germplasm yet. It'd be the challenge there. Yeah.
I guess before your breeding program started, there was no such thing as hybrid condiment mustards here. So there's varieties now that are of brown that are hybrids. There's sort of a synthetic hybrid in yellow, and Oriental is still under development. Where are you at with hybrids that have provided much better yield?
For brown mustard, I think the hybrid variety with high yield potential happy. For orange mustard, we would like to register hybrid varieties with at least 10% yield increase. We had a candidate hybrid variety with 10% higher yield than the checker variety Catalans, which received support for registration from the PRCO in 2024. So we'll still test the yield potential of this candidate orange mustard hybrid variety, and to make sure that the yield potential is there, and by testing multi-years and multi-locations. And for yellow mustard, because yellow mustard is self-incompatible and is all cross-pollinated species, and there is no hybrid system. And also because of the self-incompatibility, so we are trying to develop synthetic lines, or we call it composite lines. So the first composite variety in yellow mustard, AAC Yellow 80, was registered in 2020. So this composite line has 80% higher yield than the population variety, such as Denti. So, yeah.
That's great progress. And I guess for people that aren't familiar with this and hear hybrid and relate that to genetically modified or GMO, this was done. These are not GMO crops or GMO varieties.
Oh, no. The hybrid system, hybrid variety is not a GMO. No.
Other breeding initiatives, how important will it be to get clubroot resistance? Obviously, clubroot has been a real problem in canola, and a great deal of breeding work there, and the varieties now have clubroot resistance, and that keeps changing as the clubroot keeps mutating. How important will clubroot resistance be in mustard, and where are you with that in the breeding program, Bifang?
Yeah, right. The clubroot resistance is a very important trait. We are aiming to also to incorporate into the condiment mustard breeding program. And there is no clubroot resistant gene source in Brassica juncea. So because the Brassica juncea is closely related to Brassica nippus, so we are using two different approach to develop clubroot resistant lines in Brassica juncea. The first approach, we are trying to transfer the clubroot resistant gene from canola Brassica nippus into Brassica juncea through interspecific crossing. So we did the crossing 2 years ago. So we're trying to use back cross breeding methods, like back crossing the Brassica juncea to the F1 hybrid 3, 4, or 5 times. So now we have clubroot resistant Brassica juncea lines. So I think our next goal will be combine the immune herbicide tolerance with the clubroot resistant gene. So maybe in the future, so we can have the clubroot resistant and also herbicide resistant hybrid varieties for the producers in Canada. And in addition to that, we're also trying to screen wild Brassica juncea germplasm of different origin. And so far, we have not identified any clubroot resistant gene yet in Brassica juncea. And for yellow mustard, because the yellow mustard is genetically distant from Brassica nippus or from Brassica juncea, so we can't use the specific gene transfer approach to develop clubroot resistant lines, to develop clubroot resistant yellow mustard. So we are aiming to screen the yellow mustard germplasm from different source, because the PGRC has a yellow mustard germplasm from the different parts of the world. So far, our preliminary screen results indicated that there might be resistant gene there. Maybe next year we will see if we have the clubroot resistant gene source in yellow mustard.
Wow.
Yeah.
Salinity. There's some work going on in trying to get some better salinity tolerance in mustard.
Salinity is as a research scientist is Raju at ABC. He's working on this part. I know that he screened different lines. There might be some differences between different lines, but then we have to talk to Raju.
Now, your work is of course supported by the Diverse Field Crop Cluster, which provides some government support, and by producer money and Mustard 21 money, industry money. Just can you explain the relationship between your breeding program and Mustard 21? Mustard 21 does more of the commercialization of registered varieties, or at one point do you hand it off?
So we we develop the variety. Once the variety is registered, and then we provide the MAS 21, for example, the brown mustard hybrid variety. So once we register the hybrid variety with CFIA, and then we provide MAS 21 with the parental line as Ugura CMS A line, B line, and a restore line for multiplication, for commercialization. So, and also before that, so we had a collaboration For example, Chile winter nursery, and so we produce test hybrids there. Yeah. And the year the trials, we have multi locations, and Master 21 contract a different private company to do the trials.
Now, I suppose the winter nursery in Chile just gives you an extra season, just helps speed up the whole process.
Yes, yes. So once we identify, for example, we had a nursery to evaluate the performance of the parental lines in Canada. So once we identify some elite parental lines, and then we can use the Chilean winter nursery to make test hybrids, and then we can evaluate the test hybrids, the performance of the test hybrids next year. So yeah.
So looking back and reflecting on the past 15 years, are you amazed by how much progress, or do you look and say, oh gosh, I wish we could have moved faster, or what are the highs and lows as you look back? It always amazes me how much work and effort it takes to make breeding progress.
I think if we're looking back, so when I took this job, so the brown and white mustard is a pedigree breeding method, and then the yellow mustard is a population improvement, and this has been done many years. So yeah, I was thinking we should probably do something different. So then we took efforts, and we worked on developing the hybrid system, and we did it. So we had a hybrid system now available, and for making hybrids in brown and orange mustard. And for the yellow mustard, and we also trying to first develop elite inbreed lines, and then to make a composite lines, and for yearly improvement, it seems worked. With the mustard industry's request on herbicide-tolerant mustard, we tried to create herbicide-tolerant germplasm by using the EMS seed mutagenesis approach. And then we are very lucky we got it. So I think we're really lucky, what we aim to achieve, and we achieved it. But because we are working on 3 different crops at the same time, and there is a limitation, see how much we can do. And
You're really working with 3 crops
yeah,
rather than one.
we're working with 3 different crops. I mean, if it's one crop, maybe we have more space, we have more people, more resources to work a little more. But I mean, it's very good, and we set up a very good stage, or laid a very good foundation for the future. So we have the system ready, and we also have the desirable traits, especially herbicide-tolerant trait and carb root resistant trait now available. So I think we can probably make more progress in the future. And for yellow mustard, we designed a different approach. It worked. So I think we can probably use this approach for the future breeding program, or we can do probably something, or maybe trying to develop the hybrid system in yellow mustard using the new technology like gene editing. Yeah.
Bifang, you've done amazing work. The mustard industry is very lucky to have you and your expertise, and I think you've had some luck along the way too. Thank you very much for joining us and chatting. Dr. Bifang Cheng, Agriculture, Agri-Food Canada.
Well, thank you. And breeding work is a teamwork. So I would like to take this opportunity to thank my team members, also the chemistry lab, also the molecular marker lab at the ABC, and also the greenhouse team and the management at ABC for their support. Thank you.
Thank you, Bifang. Bifang with Agriculture and Agri-Food Canada, which is part of the Diverse Field Crop Cluster. I'm Kevin Hirsch. Thanks very much for listening.